Hi, welcome to Chapter 10. We need to talk about Mendel and some of these models of inheritance. So Gregor Mendel, right here, developed some of the fundamental laws of heredity many years before we knew this was, or we could tell with a microscope and other experiments that this was true. So here is Gregor Mendel, and these are some of the plants that he worked with. So basically he was a mathematician and he was a monk, and he decided to do some experiments on pea plants. And with these pea plants, he was looking at the traits. What makes a plant tall? What makes a plant short? Or what makes the seeds round or wrinkled? What kind of flowers do they have? And basically, he came up with something called the particulate theory of inheritance, based on there were some sort of minute particles and that these were hereditary units. They were passed from one plant to another. Well, we know this as DNA. We know that DNA is copied into RNA, that RNA expresses proteins, and those proteins would determine what color flower you are, whether your seeds are wrinkled or round, if you're tall or short. So he used his understanding of mathematics and his careful record keeping in order to come up with a theory many years before its time. So monohybrid crosses, this is what we're going to look at first. Basically, if we're just looking at one trait, so we're going to start with simple, and then we're going to move our way to a little bit more difficult. You might see this F1, F2 generation. It just means first or second and filial generation. That's why it's called F. All right, so this is they give them letter designations. So T means you're going to be tall, and a little t means you're going to be short. But the only way you can be short is if you have two of these genes, because the T is dominant. So basically, if you have one T, you're going to be tall. You have to have two T's in order to be short. So if you're big T, you're going to be tall. If you're big T, you're going to be tall. If you're little t, you're going to be short. So he took two plants, a tall plant and a short plant, and he bred them, and he ended up with a whole bunch of plants that were big T, little t. Well, this parent can give the big T or the big T, and this parent can give the little t or the little t, but that's it. That's the only thing they have to offer their offspring. So all of the first generation ended up tall. Remember, I said you only have to have one T to be tall. This is called a recessive, or you can think of it as a hidden trait. But then he took two of these and crossed them together. This parent either donated their big T or their little t, and this parent either donated their big T or their little t. So he came out with 3 tall plants and 1 short plant when he was breeding them. So he said there must be something that's passed from trait to trait. How did we get a short plant if both parents were tall? There had to have been something hidden in their genome. So what's interesting is he put together these laws of segregation, and what's funny is we just went over this with mitosis and meiosis. We know this to be true. Each individual has 2 factors for each trait. Well, what do we know? We each have 2 chromosomes for each trait. The factors separate during the formation of gametes. Well, what do we know happens? They might copy themselves, but eventually we have 1 in egg and 1 in sperm, depending on male or female. Each gamete contains only 1 factor from each pair. What do we mean by gametes? We mean egg or sperm. So each factor is either egg or sperm. Fertilization gives rise to a new individual with 2 factors. Well, if we take the 1 factor and the 1 factor and we put them together, we now have 2. So we know all of this to be true, and Mendel came up with the law of segregation. So here's back to my dominant and my recessive allele. So a dominant allele masks or hides the expression of the other allele, which is recessive. We also have some other words. We can talk about genotype versus phenotype. So genotype versus phenotype. This refers to the genetics. Big T, little t, big T, little t, the genetics. Phenotype has to do with physical. Was it tall or was it short? And then we use words homo or heterozygous. So homo means same, hetero means different. So when you look at this table, you can see big T would be homozygous dominant, and the plant would be tall phenotype. Big T we now call this heterozygous, but we still have a tall plant. And then little t we call homozygous recessive, and it's short. So you can see that this is more specific and this is more general, because these two plants are different, but they don't look different, right? Genetically they're different, but physically they don't look any different. So here might be a better example to show you some of the chromosomes, some of the homologs. So let's say these are both chromosome 1. This one came from mom. This one came from dad. But they have different alleles here. So mom has blonde hair, dad has brown hair. Now what happens to these chromosomes? Mom copies herself, dad copies herself, and now we have sister chromatids, and we also call these homologs, right? So this is called a dihybrid cross. Why is it di? Because we're looking at two traits. So they have an F1 generation of big T, big G, little g. And G has to do with green, and T has to do with tall. These are the possible outcomes if we mated two of these. So we have this individual here, and here we have this individual. So these are sperm and these are eggs. And if we take all the possible combinations, these are the outcomes we're going to get. And so in a dihybrid, you usually see this ratio. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 that are tall with green pods. We have 1, 2, 3. 3 which are tall with yellow pods. We have 2 that are short with green pods, and we have 1 that's short with yellow. Again, this is the recessive, and you see the recessive, you have to have 2 of them in order for that gene to be expressed. So if you have a big G, little g, you're going to still end up with green. If you have a little g, then you're going to get a yellow. So one of the best ways to look at how you come up with the possible gametes is something called first, last, inner, outer, or some people use the FOIL method from math. So first, outer, inner, last. However you want to look at it. So what they're saying is, how did I figure out what the gametes would be for this? How did I know that the gametes would be these? So big T, big G, big T, little g, big G, little t, little g. These are going to be your gametes. These go down, and then you put these gametes here, and they go across. So you see how you have big T, big G, big G. So what is your offspring? Big T, big G, big G. Next I want to mention something called a test cross. Sometimes you don't know what an individual is, what their genome is. So let's just say, in this case, I'm going to cross this out, and I'm going to put a question mark. If you're a tall plant, your genotype can be what? This means you're tall, and this means you're tall, right? Well, let's say we don't know. We're just looking at the plant. We aren't able to do sequencing. We're just looking at it, and he's tall. Well, this one is short, and remember, the only way you can be short is if you have 2. So we know the genetics behind the short plant, but we just don't know what the tall plant is. So if you get offspring that are both tall and short, so half and half, Remember, one chromosome has to go to either the egg or the sperm. Well, this one, it can only give a little t, right? This, we don't know what it is. But what happens if we end up with a short child? This is offspring, right? If we know this plant gave one of the T's, where did the other one have to come from? The other one had to come from the tall plant. So that means the tall plant has to have this genome. That's the only way you can get a short offspring. What about if the plant is big T, big T? So again, let's say we don't know what this is. We just know this is tall, so we have no idea whether it's big T, or big T, little t. What happens if every single plant that you get is a mixture? It's a big T, little t. Everybody is tall. If everybody is tall, what's the chances that this tall plant is actually big T, big T? It's pretty good, right? It never had a little t to give. That's why there are no short plants. So how do we apply this to humans? Well, we can look at, let's say, earlobes, or you can look at widow's peaks, or you can look at diseases as they travel, you can say, throughout families or pass down through families. So in some of these cases, so here are two people that have free earlobes. They call it free. And you can see this one, it's attached to the side, right? So basically what you do is you take these two parents and you split it down the middle, because you know this parent can either give big E or little e to their egg, and this one can give big E or little e to its sperm. So then these letters go down and these letters go across. So this is big E, this is big E, little e, this is big E, little e, this is little e, little e. So you will end up usually, if you take two parents that are heterozygous and you mate these two heterozygous parents, you usually end up with this 3-to-1 ratio where one recessive is going to show up. So we can also use this if we're going to talk about recessive disorders within a family. So here's a person that has a disorder. The recessive disorder, having two recessive alleles gives you the disease. And you have a parent over here, and say we don't know what his genetics is. This is where those kind of test cross come in. You can look at the family tree. Well, they had two normal children, and then they mated and had another child, and their child was normal. But these two are related. When these two had children, their children were all normal, but then eventually we have these autosomal recessive disorders. And this is a little weird family tree because these were relatives, right? So these were sisters, and so then this would be first cousins, right? And then they had children. But anyway, so you can see how the disease is passed down through families. And this is an autosomal dominant disorder. So all you have to do is have one of the dominant alleles and you're going to have the disease. And you can see it's present in every generation because it is dominant. Where you can see with the recessive, it's skipped 2 generations, and then now we see it prevalent in this 4th generation. So beyond Mendel's laws, we have to talk about some other types of inheritance. It's beyond the scope of what he saw when he was dealing with plants. There are some other types of inheritance. So this one is called incomplete dominance. What happens is the phenotype is the same as the genotype. It's reflective. So let's say you have a pink plant. So this is a pink plant. And think of R1 and R2, same thing as big T, little t, except for now we're trying to say color R1, R2. So if these are on, remember these are on chromosomes, so this would be R1 and this would be R2, we don't know which gamete is going to get which chromosome. So R1 or R2 are the possible egg combinations, and R1 and R2 are the possible sperm combinations. What we're doing is we're taking an R1, R2 plant and we're mating it with an R1, R2 plant. So this plant is red, these two are pink, and this one is the recessive white. So what do you notice? If you have two R1s, you're red. If you have R1, R2, meaning you're heterozygous, you're pink. And if you're recessive, R2, R2, you're white. There's also something called multiple alleles, and blood types are determined by multiple alleles. This might be a little confusing, so I'm going to kind of use this AB, and O type. So on all of your chromosomes, you have two. Let's say this, you have A, and this you have A type blood. Basically, they're tags on your red blood cells. That's what these genes code for. They make a protein that's expressed on your red blood cells. So you can be AA, or you can be AO. Both of these are going to give you A blood. Again, this is recessive, so it's hidden. Same thing as big T, big T, big T, little t. These were both tall, right? But what happens if you have one chromosome that has A and one chromosome that has B? You end up with a blood type called AB. It is a multiple allele. There are several forms, and depending on which forms of the allele or which part of the gene you have, you'll end up with a different blood type. The O is recessive, the OO. That's because I'm already thinking about chromosomes. The O is recessive, so that means you have to have two O's. Just like when we talked about short, you had to have two T's to be short. Well, the O is the recessive. So that's an example of multiple alleles. Codominance is how they explain AB. So this makes a protein and this makes a protein. They both get expressed on your red blood cell, and so they are both fully expressed. They call it codominance. There's also something called pleiotropy. I just want to mention it, especially when we talk about sickle cell. We've talked about sickle cell disease, when a single gene has more than one effect. So sickle cell disease has to do with red blood cells, but because of their shape, it can cause anemia, cause weakness, poor circulation, because the red blood cells, remember, they were sickled and they can get stuck, causes pain, causes spleen damage and kidney damage because your blood goes through these organs in order to be cleansed. There's something called Marfan syndrome, and it has to do with connective tissue, and connective tissue is throughout the body. So if you have abnormal connective tissue, then that means any part of your body that utilizes connective tissue, it's going to have an issue or a problem. There's also something called a polygenic inheritance, and I kind of wanted to show you this picture so that you could see when it comes to skin color. We have, let's say, light alleles, and we have dark. So the more of these that you have, the darker your skin will be. The more of these you have, the lighter your skin will be. So if we take this first generation, and two people, they have 3 and 3 of each, right? And we mate those, the combinations, that the possible combinations of how these chromosomes get split up during meiosis 1 and 2, these are all the possible combinations that you can have for offspring. And you can see the more, if you have an increase in dominant alleles, you're going to have an increase, that's hard to write, increase in skin pigment. So this is darker skin, this is lighter skin. So it has to do with the polygenic inheritance of pigment or skin color. There's also something called sex-linked inheritance. Basically, there are some genes that are on the X chromosome, and they can be passed down from mother to child just like they can if they're on a regular chromosome. So now the only difference is we're talking just about X and Y chromosomes. So in these, they're trying to show you white-eyed flies versus red-eyed flies. So this has red eyes, it is dominant, and this is homozygous, right? This is a male, and this is a female. Well, if it's carried on the X, the female has 2 X's, but if it's carried on the X for the male, then he only has 1 X. So if he gets the only one recessive, he's going to have white eyes. he doesn't have another X with a dominant to mask the recessive. So basically the recessive shows in the male because he doesn't have another X. So they're now saying, let's mate these two flies. We're going to have a white-eyed male fly and a red-eyed female fly. Now she can only give her red dominant allele, right? She has two of them. That's all she can give to her offspring. Well, the male can either give the Y or he can give the recessive white-eyed gene. But if we're talking about a male or a female, it depends on what they get. So let's say this X goes over here and this X goes here. So you end up with a female that has red eyes, but you see she has the recessive allele, recessive white-eyed allele. The male fly, he got his Y from dad, so he can't get the X. He got the Y from dad, so he gets this X from female, his mom. Now they're going to mate these two together. So here's the XR, big R, here's the X little r, here's the big XR, and here's the Y. So when you do your Punnett square that these go down and these go across, you end up with a red female, you end up with another red female, you end up with a red male, and you end up with a white male. The only thing that's different about this is when you list phenotypic or genotypic. So how we have two red-eye female, we have one red-eye male, and we have one white-eye male. So you have to differentiate between male and female. And then if you wanted to, genetically, if you wanted to do the genotype, I'm adding extra letters today, you'll have a, what is this, homozygous, dominant. Then you'll have a heterozygous female. So this is female, this is female. This is a homozygous red-eyed female. This is a heterozygous red-eyed female. This is a red-eyed male, and this is a white-eyed male. So you could also have genotype in there as well, but you do need to note male versus female. Again, how can we use these when we talk about following family trees or pedigrees, looking at X-linked recessive disorders and how they're carried from one family to the next? All right, I'll see you for the next chapter. Bye.